Zhangwei.
Good afternoon, Jennifer, and welcome everybody to episode 7 of Making Reproductive Longevity a Reality. So I'm Zhongwei here. I'm a clinician scientist in obstetrics and gynecology, and my research interest and focus is on trying to change the irrevocability of reproductive aging in women, hopefully for reproductive longevity.
I'm Jennifer Garrison. I'm a faculty member at the Buck Institute in Novato, California. I'm also a member of the Center for Reproductive Longevity and Equality here at the Buck, and the faculty director for the Global Consortium for Reproductive Longevity and Equality. And that is a mouthful. But the consortium is really designed to build this global ecosystem around female reproductive health and equality. And we're doing that through funding, through collaboration, through dialogue between scientists like me and clinicians like Zhangwei. And what we want to do is create opportunities for interaction and collaboration that cross cultural and physical divides to remove barriers to progress in this field. And this webinar series is really designed to highlight some of the research that we funded. So today we're featuring 2 talks from GCRLE grantees on really important and exciting topics related to reproductive aging. Namely the role of the gut microbiome in ovarian dysfunction. Now, we did hear about that on a previous webinar, but we're going to continue talking about that today, and how oocyte or egg quality might serve as an overall marker of a woman's health. So, we know that ovaries age prematurely at nearly double the rate of tissues in the rest of the body. And the end of fertility or the entering into menopause really sets off a negative cascade of health effects in a woman's body, even a healthy woman's body. It puts a woman at risk for cognitive decline, for cardiovascular disease, for stroke, just to name a few. And we think of ovaries as the so-called canary in the coal mine for aging. And keeping them in a youthful state might actually help improve overall health during aging. So today, we're going to talk first about how microorganisms in your gut might influence how ovaries age. Then second, we're going to talk about how mitochondria, which are like the batteries of a cell, they actually provide the energy, how their performance and how well they're pumping out energy inside oocytes might actually influence overall health and maybe even predict how a woman ages in other tissues. So Zhangwei, you probably talk to a lot of women in your practice who are experience the negative health effects of going through menopause.
Yeah, actually many of them ask me before they even hit there, how do we maintain the youthfulness and not to be affected, by this impending menopause, many say. And can I do something about it? Can I eat something? Is there any supplement? Is there anything that's advice for them? Because all I can tell them now is that I'm sorry, the important organs that you never see in your life, which is your ovaries, are actually controlling everything. And somehow they are aging before any part of your body system. And today's topics are so interesting because perhaps you could find something, that I could change. What I'm going to tell this lady, could this science behind all these solutions to keep their ovaries young and to keep their healthy lifespan, for these women longer and more productive and, greater happiness, isn't it? Ladies?
Yeah. Yeah, exactly. So we're going to reserve the questions to the end as we have in the past. So if you have questions, please put them in the Q&A box and we will get to them at the end. We'll have a discussion with both speakers. And I am delighted to introduce our first speaker, Minhoo Kim. She is a postdoctoral fellow in the Benayoun lab at the USC Leonard Davis School of Gerontology. Minhoo completed her undergraduate and her graduate studies in South Korea at the Korea Advanced Institute of Science and Technology, or KAIST. Her PhD research in Daeop Lee's lab focused on understanding how mRNA export in yeast actually can influence stress in order to maximize cell survival. And her interest in genomics and aging biology led her to Berenice's lab at USC. And remember, we heard from Dr. Berenice Benayoun in week 4. And she's really got a really extensive expertise in genomics and mammalian ovarian aging. And so as a postdoc, Minhoo is getting trained in this field, and she wants to investigate how there's a bidirectional relationship between the gut microbiome, or the microorganisms that reside in your gut, and ovarian aging. So today she's going to tell us about her project in this area, and I'm very excited. So welcome, Minhoo.
Thank you for the kind introduction. So again, my name is Minho Kim. And before I start my presentation, I would like to thank the organizers from the National University of Singapore and GCRLE, especially Jennifer and Bevan, for this amazing opportunity to discuss my research field. So today I will be presenting on my project, which aims to understand the causal role of the gut microbiota on female reproductive aging. And hopefully by the end of my presentation, I would have convinced you all that the gut microbiota has a widespread role, not just in female reproductive aging, but on host well-being and health. The microbiota is defined as microorganisms that live on the surface of the host. So these microorganisms include bacteria, fungi, and viruses, and these all can be commensal, symbiotic, and pathogenic. So basically meaning they can be both good or bad to the host. Microorganisms are found on multiple surfaces of the host, such as the oral cavity, skin, digestive tract, or gut, and urogenital organs. As I'll be getting to throughout my presentation, the microbiota has been shown to have a significant effect of the health of the host. It has been referred to as the forgotten organ. Among the different microbiotas, the gut microbiota has been the most extensively studied. And one of the reasons for that is because of its close relationship with the immune system of the host. So in case of human, the adult human gut microbiota is said to be composed of about 10 to 100 trillion microorganisms. And so far, thousands of different species of these microbes have been identified. And this number is actually 10-fold more than the human cells. So just to get a perspective of this number, I did a brief Google search. And apparently for a 70-kilogram human adult, about the gut microbiota would weigh about 200 grams, which is equivalent to your mid-sized mango. Nonetheless, what's interesting about the gut microbiota is that its composition undergoes significant changes throughout lifespan of the host in response to various factors, including lifestyle such as exercise, diet, stress, medication, age, and host genetics, just to name a few. So what we are interested in is that whether these microbiota, which exist in such great numbers, can have an effect on the health of the host. So in 1907, Eli Mechnikov actually proposed that age-related dysfunction results from increased colon permeability-driven chronic systemic inflammation. So that's a mouthful, but to understand that, we have to understand the two contrasting states of the gut. So in gut homeostasis, the gut microbiota and host immune system are in equilibrium. So the cells aligning the gut barrier are intact, and it keeps the pathogenic microbes from intruding into the host. So they are in constant communication to ensure this homeostatic state. Whereas in gut dysbiosis, which may result from disease or just deterioration due to aging, this gut homeostasis is lost, and the cells aligning are no longer functioning as a barrier, which leads to this leaky gut state and overall induces chronic systemic inflammation in the host. So going back to what Eli Mechnikov was proposing, he was suggesting that the dysfunctions that are associated with aging results from this leaky gut state, which ultimately leads to a systemic inflammation. As a side note, actually he also proposed that we can lengthen our lifespan by consuming healthy bacteria in yogurt, specifically lactic acid bacteria. That's why we have this comical image of elderly people drinking yogurt out from this fountain. But later, studies have shown that it's not as simple as that. So in any case, thanks to the advancement of different techniques and analytical tools, studies have actually showed that microbiota can affect the host health and well-being significantly. Excuse me. For example, the gut microbiota is important for metabolism, including food degradation, lipid storage, and vitamin synthesis. And metabolites, micro or macromolecules that are produced by the gut microbiota, is required for multiple cellular processes. So concurrently, this unhealthy gut microbiota or dysbiosis state has been attributed to pathogenesis of metabolic disorders such as obesity, diabetes, and more. Rather unexpectedly, the gut microbiota has also been shown to regulate brain function, which led to coining of the term gut-brain axis. So briefly, through production of those metabolites or through direct interaction with the neurons that align the barrier of the intestine, the gut microbiota can cause the development of multiple sclerosis, depression, and anxiety, just to name a few. And last but not least, very recently, it was shown that the gut microbiota can even regulate further lifespan. So all these studies strongly suggest a direct role of the gut microbiota on overall host well-being. But relevant to my research interests, multiple studies have also shown the effects of the gut microbiota on ovarian function. So in 1978, Dr. Lombardi showed that human fecal microbiota is capable of metabolizing estrogen, which led to coining of the term estrobolome, which refers to the collection of gut microbes capable of metabolizing and modulating the body's circulating estrogen. And also studies have shown that grafting or transplanting different concentrations of human fecal microbiota can actually alter circulating serum estrogen levels in mice. Also, very recently, a study showed that the gut microbiota is required for proper sexual maturation in mice. And on the right, I brought a representative figure from this paper, which was very recently published, that demonstrated that. So we are looking at a histology data of ovarian tissues that were collected from conventionally raised mice, so normal, healthy mice. And on the right, we are looking at germ-free mice, which are referring to mice that were raised in these facilities that allow elimination of natural microbes that reside in your animals. So when we look at the normally raised, conventionally raised mice, we see a well-organized structure of these theca cells around the granulosa cell, which is expected in a healthy ovary. Whereas on the right, these germ-free mice actually present with this disorganization of theca cells around the granulosa cells. So obviously this paper did not rely on this one single data to come up with this conclusion, but overall, this paper showed that gut microbiota is actually very important for proper sexual maturation in mice. And last but not least, in another very recent study, it was shown that transfer of fecal microbiota from PCOS, polycystic ovarian syndrome patients, disrupts mice ovarian function. So PCOS is a condition that is observed in relatively younger females that is due to hormonal imbalance, and it results in these cystic structures in their ovaries and in fertility. So what they did in this paper was that they transplanted these fecal microbiota from PCOS patients into these recipient mice. And when we take a look at these histology data of ovarian tissue comparing control versus PCOS fecal microbiota transplanted mice, we see on the left average expected number of corpus lutea, which are a mass of cells that are required for menstrual cycling and early pregnancy. In the PCOS fecal microbiota transplanted mice, the number of corpus lutea not only decreased, but they also observed these cystic structures in the ovary, which is equivalent to what we see in human patients. On the right, they also did a fertility assay where they counted the number of pups that were born from these recipient mice. And as you can see on the right, the number of pups that were born from the PCOS fecal microbiota transplanted mice were significantly lower compared to their control group. So overall, these studies suggest that the gut microbiota can, in fact, have a direct role on ovarian function. So based on this, my study aims to understand the effect of gut microbiota on female reproductive aging, so onset and progression of menopause. To do that, my study is set up in 3 different steps. So microbiota collection, microbiota transplantation, and data analysis. So in the first step, we will be collecting fecal microbiota from 3 different states of menopause from this donor mice. So premenopausal, peri, and postmenopausal states. And for our second part of the project, we will be grafting these fecal microbiota onto these recipient mice that were depleted of their original microbiota by feeding them with antibiotics water. Then after, we will be testing these recipient mice for their changes in their ovarian landscapes through histology analysis, serum analysis, and others, as well as analyzing any changes in the microbial profiles in these recipient mice. So through this study, we expect to identify microbial remodeling events and candidate metabolic pathways that contribute to menopausal progression. And also we hope that findings from these studies will provide new opportunities in developing methods to delay ovarian aging or mitigate its effects. So with that, I would like to thank my amazing supervisor and mentor, Berenice Benayoun, and the members of my lab for being very supportive and helpful in structuring this project and moving forward. And thank you very much for your attention.
Thank you, Min-Hoo. I think that it's really fascinating. I mean, I already have a few questions in mind, but before we go on to discussion, allow me to now introduce my fellow physician scientist, Lynae. She's a physician scientist with clinical training obstetrics and gynecology and reproductive endocrinology and infertility. Her basic research focuses on using the oocyte as a marker for overall health, with a specific emphasis on how oocyte mitochondrial physiology could predict somatic aging and long-term health sequelae. And the clinical research also focuses on femtech as a mechanism for sexual health education and period tracking for early insights and health. So Lynae, can you please now shed some light on how the oocyte can give us a marker of general health in women? Lynae, please.
Thank you so much for this opportunity. I'm going to start sharing my screen. Wonderful. So I believe that the oocyte is a crystal ball. It can provide us a marker for overall health. And I'm just going to divide my talk into 4 sections. I'm going to give you a little history of IVF and infertility and a condition called diminished ovarian reserve. I would like to talk about oocyte mitochondria, poor oocyte quality and mitochondrial physiology with some data from my lab, and also the long-term health sequelae associated with poor oocyte quality. So traditionally, in many cultures around the world, fertility has been important. And I'm showing you pictures all of different fertility goddesses around the world. Unfortunately, the progression in reproductive biology has been quite slow. With the publication from William Harvey in 1651, he declared at ex ovo omnia, which means all things come from the egg. And unfortunately, that's where the field remained for many, many years. It wasn't until 1978 that we were able to capitalize on some of the information from sea stars and sea urchins that there could be external fertilization, in vitro fertilization, with the birth of Louise Brown, the first IVF baby in 1978. However, we haven't gone far enough to understand really the underlying causes of infertility as it relates to the oocyte. Yet 1 in 6 couples worldwide suffer from infertility, and nearly 10% of women worldwide will have infertility for at least 12 months of their life. And so it's a significant health problem. And infertility is defined as the failure to achieve pregnancy within 12 months of unprotected intercourse or 12 months of therapeutic donor insemination. And this is actually a shorter time, 6 months, in women who are older than 35 years old, because that is advanced maternal age. And of course, fertility decreases, with age. And so this is a graphic that I like to show because essentially as your fertility is decreasing in your mid-30s when you become advanced maternal age, your actual rate of miscarriage increases. And so even if you're able to attain pregnancy, the chances that you'll have a live birth decrease as you age. However, we know with individuals who go through in vitro fertilization and use donor egg from another individual, they actually don't have a decrease in live birth, regardless of age, because everything hinges on the oocyte. However, if you use autologous eggs, you see that the live birth rate decreases dramatically in the early 40s and almost to nothing in the mid 40s and late 40s. This is an overview of IVF. So in panel A, you can see here, this is an ovary that is not stimulated with the white arrow pointing to it. However, after gonadotropins, the ovary is enlarged and you can actually see individual follicles. And what we do with IVF is the patient is anesthetized, a probe is inserted into the vagina with a needle, and the needle actually goes into the ovary and aspirates these individual follicles. And hopefully there'll be an oocyte there that is retrieved that will be fertilized either through traditional IVF or with ICSI, which is cytoplasmic sperm injection, and then the embryo will be transferred later, hopefully when it's a blastocyst seen in panel H here. And this is a close-up cartoon of what it looks like here on the left. And here's an actual ultrasound with a needle going into a follicle to aspirate it. However, we find that a lot of patients who go through IVF have something called diminished ovarian reserve. And unfortunately, there's no uniform definition, but it essentially means that there's low oocyte quality, there's low oocyte quantity, and that there's reduced reproductive potential. And unfortunately, we don't know the cause of diminished ovarian reserve for most of the cases. The American Society for Reproductive Medicine does keep information about IVF cycles, and we know that about 32% of all in vitro fertilization cycles in the US have a diagnosis of diminished ovarian reserve. So it's quite common, yet we don't have solutions. However, there is a theory about oocyte quality that stems from the mitochondria. This was published, a review by Mai Panlu et al. In 2016, which suggests that mitochondrial dysfunction actually hastens or precipitates oocyte aging or ovarian aging. And the mitochondria are very important to the oocyte. There's about 100,000 mature, there's 100,000 mitochondria in mature oocytes at the time of fertilization. And yes, they do make ATP, but they do other things. They all come from the mother because they all come from the oocyte. They're maternally inherited. They also direct calcium homeostasis. They direct apoptosis, which is really important to the ovary. And of course, what I want to highlight here is also fatty acid metabolism. The mitochondria increase as the oocyte develops. So again, there's 100,000, but however, mitochondria are different between different cells within the same animal. On the left panel here, I'm showing you oocyte mitochondria. This is taken from my lab, from my mice. And this, in the same mouse, the oocyte mitochondria are very different compared to the kidney mitochondria. They look morphologically differently, and they also probably function differently. And what's important is that I wasn't really interested in mitochondria before, but I was interested in how the oocyte can protect itself. And so I was working with a plasma membrane transporter called multi-drug resistance transporters. And so I was interested in how these transporters, which efflux toxicants, namely in the gut as well, and in the blood-brain barrier, how these toxicants could work together potentially to protect the oocyte from chemotoxicants such as chemotherapy. And so my very first foray into science was from a clinical question that I had was, is MDR1 or multi-drug resistance transporter 1 expressed in mammalian oocytes? And I found this out with my very first publication that yes, indeed, in the murine and human oocytes, it's expressed. And we found that it's really important in protecting against chemotherapeutics. And so I started to wonder, was there a knockout or a mutant that I could work with to understand what happens when it is not functional? And so we worked with a mutant that has the protein, but doesn't have a functional protein because part of its ATP binding domain is actually dysfunctional versus the knockout, which has no protein. And we found that this mutant had these global transcriptomic changes with an upregulation of genes that are related to metabolism and mitochondria. And so I actually did a very simple assay where I took the ovaries from these mutants and looked at the metabolites that were found in the ovaries. And this is a graphical representation of what it looked like. It looked like there was an accumulation of metabolites that were involved in the TCA cycle. And I wondered, what does this have to do with protecting the oocyte? But I started to see more data that actually the MDR1 and mitochondria likely were interrelated. And so we did a very simple thing with immunofluorescence and Western blot to see if there was any co-localization. And lo and behold, there is. So MDR1 is expressed in the mitochondrial membrane. And that was really exciting for us because they had not been shown in the oocyte before. And what we found were that the MDR1A mutants who have oocytes, GV, germinal vesicle oocytes, which are immature oocytes within the ovary, had increased reactive oxygen species. And so that was something that we were not anticipating. And so this ROS, or reactive oxygen species, can be deleterious to not only the mitochondria, but the overall metabolism. And we found that when we did both MitoSox and MitoTracker, we saw that essentially the ROS was really important and was consistent. And then we also submitted this paper and the reviewers wanted us to look at cellular ROS as well, not just mitochondrial ROS. And we also found that the mutants had more cellular ROS as well as mitochondrial ROS. And when we looked at the membrane potential, which actually is an indicative marker of mitochondrial health and its ability to make ATP, we found that the membrane potential was reduced in the mutant, mirroring the positive control. And so this was really exciting for us because it showed that perhaps the physiology was aberrant. We also looked at morphology between wild-type mice and the mutant mice. And the mitochondria in the mutant showed morphologically distinct features from the wild-type, suggesting that they were perhaps compensating for some physiological defect. And specifically about their fertility and their oocyte quality, we found that the wild type versus the mutants, and the mutants, we found that with natural ovulation and with superovulation, they made more oocytes, which seems good at first, but actually in reality, the oocytes lysed very easily with manipulation. And even though they made more oocytes, they didn't make more pups. So they didn't have more babies. And if you look at the panels below here, the wild type ovaries have a very consistent look. But our mutant ovaries always looked hyperemic. They looked larger. In fact, they always looked like they were superovulated when they weren't. And you can see what a superovulated wild type ovary looks like here versus a superovulated mutant ovary here. And so we actually aged our animals because we weren't sure what to make of this. And when we aged our mutants, we saw that they ovulate less oocytes as they age. And this is what we see with diminished ovarian reserve in human patients. However, there is a link here. My former postdoc noticed that the babies, the pups, born to our mutants were actually heavier. They're eating the same food, they're in the same conditions, but they were heavier at postnatal day 21 as well as postnatal day 42. And that continued into adulthood. So the females were heavier as well as the males. and they had larger livers. And you all know clinically, enlarged livers potentially could suggest fatty liver disease. And so we looked at oil red O staining to look at lipid content within the liver. But unfortunately, I'm a reproductive endocrinologist, and this was really not in my wheelhouse. And so I started to think, this is a canary in the coal mine. Perhaps the oocyte quality is associated with some type of metabolic dysfunction. And so I needed a little bit more help, did a little bit of research, and there's a lot of data that says that lipotoxicity basically decreases oocyte yield, that ROS actually is increased in obese mice, and that mitochondria have disrupted architecture, and adipose tissue is a very inflammatory organ, in fact. And so I started to look around, and here in Germany, where I'm located in Berlin, actually in Munich, there is a German mouse clinic that actually phenotypes mice. And so I submitted a proposal to this collaborative entity, and they accepted my proposal. And essentially what they do is they will import the strains of mice, they will look at the cohorts, and they will phenotype them. And this is just a pipeline overview of all of the different things that they will do to my colony along with the wild type to test them. And these are the really ones, the important ones that I want to focus on. They will be looking at overall general chemistry, glucose metabolism, energy metabolism, and also fat mass, which is really important to me, as well as flow cytometry within the spleen. And now they're going to try something new within the ovary. And that's really important to us because we want to know if this is a really good model for humans, because we know that diminished ovarian reserve does have cardiovascular sequelae. And so DOR is associated with preeclampsia. So if you take higher doses of recombinant FSH when you're going through an IVF cycle, you're more likely to have preeclampsia, which is essentially a manifestation of cardiovascular disease before it actually occurs. And we know that patients who have low AMH, meaning that they have low ovarian reserve, typically have higher cardiometabolic risk factors. And so the last point that I want to leave you with clinically is that those who have diminished ovarian reserve have low bone density. And we know that this is a hallmark sign that occurs in possibly early in the menopause or very late in the menopause, depending on the condition of the patient when they arrive at menopause. And the average age of menopause is 51.4. So the conclusion is that MDR1 is expressed in the oocyte mitochondrial membrane. And we know that just having a mutation in MDR1 can impact mitochondrial physiology in the oocyte. And we know that essentially the loss of MDR1 is associated with poor oocyte quality. But now we're trying to establish if there's an increased risk for obesity, fatty liver, or dyslipidemia, and that can inform long-term health sequelae in women who are infertile. And so we will be applying for a grant in the near future, and these will be our aims. And I thank you so much for the ability to speak to you all and for the funding from the GCRLE.
Thank you, Lynae. I think that was amazing. Yeah, thanks. And I think thank you for being up and around till about, what time is it now in Berlin? Is it 3 a.m.?
It's 2:36 a.m.
Thank you for still being so awake and still able to show so much interesting data. Jennifer, any questions?
Yeah. Oh, there are questions in the chat box, and I have questions. I'm sure you also have questions.
Indeed.
The first question that came in through the Q&A box is for Minhu. Why did you choose to transplant the microbiota in 4-month-old mice? And do you have any plans to do it with older animals?
Yes, that's actually a very good question and comment. So the reason why we chose 4-month-old is very simple. So we wanted to test the fecal microbiota, the effect of fecal microbiota on young adults. So the range people typically think of young adults in rodents, mice in this case would be between 3 to 6 months old. So we chose a 4-month-old, which is a usually used age in the lab. And the comment, if we were planning on testing it at older ages, yes. So it was part of our future plan. So if the opportunity arises, we would very like to test for the positive effect of fecal microbiota from young females to the older females.
Yeah, that'll be really exciting to see. I had a question actually related to that. There are other microbiomes in our body, right? So skin microbiome, vaginal microbiome, there are microorganisms everywhere, and they have their own communities in different parts of our bodies. Do you know if there are any effects of other microbiomes on female reproductive aging?
Unfortunately, there hasn't been a thorough investigation on a specific microbiota that is other than gut microbiota that has an effect on female reproductive aging. Although it is obvious that microbiota from your oral cavity sort of flows through your intestine and it gets mixed up in the gut. So there has been studies that suggest sort of this interplay between different microbiota, but there hasn't, if I'm correct, there hasn't been a specific study that looked at a different microbiota.
Right. I think to leverage on that before I disturb Lynae, with regards to other questions, Minhu, can I just find out, do you expect any change in the type of microbiota based on estrous cycles in the mice? Because we know in women, right? I think Lynae will know, lactobacilli is actually more abundant and is protective for women during the reproductive ages because of high estrogen levels. Do you expect that to affect the microbiota and maybe reproductive aging? Because, what is like chicken egg question? Like, what is happening? Is it the gut causing the sex steroids to, happen, or what do you think is happening at that level?
That is a very interesting question. It is something that I'm very interested in. It's just technically tracking the estrous cycle in mice is very difficult. And I see, I think my supervisor is actually in the audience, and I almost can hear her laughing when I say this, but it's technically very difficult to track the specific cyclic state the mice are in. But your question is something that I'm very interested in. It'll be very exciting to see the direct interplay between the gut microbiota and the hormone levels in the mice, depending on the cycles.
Yeah, I mean, I've been also doing some vaginal swabs of these mice myself to check their estrous cycles and whatnot. So I was thinking, could it just be a surrogate marker, for your mice? I'm sure, Lynae, will do your IVF on mice as well. Yeah, you'll probably check when they ovulate and whatnot. So I thought that was quite interesting because there's another question that was actually asking on that, which is there a particular type of microbiome do you think, is going to affect, reproductive aging? Is it just any cell microbiome, or is there a particular species talking about?
So that's the goal of our project that we are moving forward with. So we aim to identify a specific species that can drive or sort of decide the onset or speed at which the menopausal state progresses. So that's the hope. For the specific species that have been identified so far, I am not aware of that.
Okay.
Do you, Lynae, I know it's late, but we really want to hear from you too. If you have any thoughts, you should jump in. I had a question, and I think it also came in through the chat in part, about these multi-drug resistance pumps. So, getting rid of toxins and xenobiotics or, things that don't belong in the cell is a major job. Like, it's a huge role, and there are a lot of these pumps. So the question is, for MDR1, does it change over time, like during aging in different tissues, particularly the ovary?
Yeah, so we actually tried to look at this because we did RNA-seq on wild-type mice for a different project, and we didn't see a tremendous change. However, in terms of gene expression, however, I wonder if the pump continues to function at the same level with changes, and that's something we haven't tested. And there is some data, and we did try to look at this, that there's an estrogen response element in the MDR1 gene, and that potentially estrogen could drive that. But it's unfortunately, it's a very hard thing to really test because we really didn't test the range of the different types of estrogens that are available. And so what we did was we tested 17 beta estradiol, and we didn't see a dramatic effect. But that doesn't mean that different estrogens like estrone and especially the estrogen of pregnancy, estriol, might not have an effect, because we know that MDR1 is present in the placenta and is very important at detoxification, on behalf of the fetus. And so there are probably many different factors and many different inducers of activity for MDR1. And the unfortunate thing is that it's mostly been studied in cancer biology and not in normal cells.
Yeah, it's so exciting. The fact that you're seeing changes in fat deposition without any changes in the amount of calories that the mice are ingesting, that really suggests that there's a change in energy homeostasis there, which obviously involves the brain, which I think is super exciting. But I'd like to ask a question of both of you. There was a question in the chat, or a statement actually in the chat. Someone pointed out that, we're talking about ovarian aging, and we're talking about, detrimental effects on women going through menopause. Someone pointed out that, women live longer than men. So, and so a question for each of you is, does the gut microbiome change in males with age, Minhoo? And Lynae, is there any difference in MDR1 expression or function in males during aging?
Should I go first?
Sure.
Yeah. So, unfortunately, the majority of the data available right now were done in males. So there have been a lot of studies that tracked changes in the gut microbiome throughout age. So we know what specific microbes are present at certain age on average, and we know the trend. So we know that there's a decrease in diversity in these microbes as we age. We have more pathogenic microbes as we age. But studies have not compared the sex differences between the two. So what's very exciting for me is that because the Benayan Lab is very interested and committed in investigating sex dimorphic differences in aging, we have the tools and we have access to all these opportunities to assess the differences in both sex and age. So it is very exciting. I am very excited that we will be able to identify different microbial species in terms of sex and age as well. So we'll have that whole view of the gut microbiota relevant to aging.
So Lynae, is MDR1 expressed in any male specific tissue, reproductive tissue?
My favorite male specific tissue is the testes. So it is there in the blood testes barrier. It's definitely in the blood-brain barrier for obviously both gender, both sexes. It's expressed, it's actually ubiquitous. So it's in the gut as well. So in fact, the knockout of the MDR1 actually has been described in the literature to have a chronic inflammation that's similar to inflammatory bowel disease. And in fact, they were the first to document that the mitochondrial dysfunction was present in the knockout. And so when I read that paper, I was really excited, of course, because I thought, wow, okay, in the gut, there's a mitochondrial deficit. I wonder in the ovary. Unfortunately, I don't know if there is a decrease in MDR1 in males as they age. Again, unfortunately, the majority of literature, if you just do a Google search on MDR1, is cancer, 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 cancer. But that's something that's really, I think, a good point, because of course I do have male mice, we need them for the sperm. And I did have a collaborator who was looking at maybe sperm abnormalities within MDR mutants and didn't find anything that was recognizable. But again, sometimes it takes several different views of testing, because when I originally started working with this mutant line, the distributor told me that there was nothing wrong with their fertility. But clearly there's some subfertility and some issues that are still being investigated.
Yeah. And I would also point out that for the men in the audience, a woman's reproductive span is directly correlated with her lifespan, meaning that the age at which a woman goes through menopause is actually an indicator of her overall lifespan. Women who go through menopause later tend to live longer on average. And you might think, oh, that's really important for my fill in the blank, friend, partner, a sister, mother, grandmother. But that also extends to their male brothers. So there's a genetic component there that, is important for everybody.
Yeah,
Yeah.
absolutely, Jennifer. So being the male currently in the panel, I think that is an excellent point that we have to understand. And one of our attendees actually mentioned that even though women tend to have reproductive aging much earlier, and have more problems, but they tend to live longer. But to me, living longer, but you have to live well, isn't it? We are talking about health span as well, not just lifespan alone. You got to live long and well. And actually a recent, I won't call it a publication, but, a media publication in Singapore, which is our newspaper, national newspaper, actually show that many of our women are living longer, but not so well. They have higher risk of hip fractures, they have osteoporosis. We have to balance that story nicely. And then comes our next question, because that's what all these ladies are asking me. And these two ladies here have to answer my question because they're all related to eating. So 3 questions for Lynae. Everyone is excited and asks you, have you studied any antioxidant therapies to improve oocyte aging? Is there a form to supplement with vitamins and oxidative stress for MDR1? Is there a way to even monitor MDR1 activity through the blood? And some also mentioned that, can high-dose coenzyme Q10 of 600 milligrams daily help with the energy production at the oocyte and help in MDR1 mutant oocytes as well? So,
oh, that's a lot. So
yes,
I'll start backwards. I'll start in reverse. So there's been some conflicting data about coenzyme Q, and I think that a lot of people do think that having a diet that is rich in antioxidants is helpful for overall mitochondrial metabolism. The question is, does that translate to the oocyte? And we don't know, right? Because essentially you have in the oocyte, you already have a pool of mitochondria that you inherited from your mother, and you inherited from your grandmother and your grandmother's grandmother. And so what I found in another project in my data is that if your grandmother is exposed to toxicants, such as a chemotherapeutic, and you don't have an intact multi-drug resistance transporter 1 pump, what happens is that the effect that grandma has goes down to mom and then granddaughter. And if you have an MDR1 mutation, if you don't have an MDR1 mutation, the effect stops at the child and is not seen in the grandchild. And this has all been in mice. So to answer the question about, is there a functional assay for MDR1 in the blood? Not that is commercially available or clinically available. One of the things that I'd like to do with my funding, thanks to the GCRLE, is that I'd like to actually try to determine what the mutation rate is in the human population, because I think that's still a question mark. There's been some data, but again, very, very little, because there's a couple papers that suggest in the US that there's a mutation rate as high as almost 25%, but it may be associated with breast cancer, it may be associated with inflammatory bowel disease, but there's not been any hard and fast data that suggests that. And so there's no assay to know what polymorphism is more deleterious, but there are known to be over 60 loss of function polymorphisms in the human population. And I think, if I could look into a crystal ball and say what's going to happen in the future, I would love to be able to have an assay that predicts someone's oocyte quality before they actually present with infertility. And that's really important, because we don't have a lot of clinical markers that we can use besides the menstrual cycle, and paying attention to the menstrual cycle as well. I think I hope I answered all the questions.
You did. I think so. Yeah. There's another question related to food and metabolism from Minhu. Does the food we eat affect our microbiota and also reproductive aging?
Yes. So diet is one of the main factors that shape our gut microbiome. So the answer to that question is yes. So food we eat can affect our microbiota. Can it affect reproductive aging? So that's the hope. So we are still at a state where we are testing out adverse effects of the microbial species that we identify in our research. So we're feeding old fecal microbiota to our young. But as Jennifer commented earlier during this Q&A session, if we were to later test out what effects the young fecal microbiota have on the older aged mice, we are hopeful that we'll be able to identify microbes that can mitigate the effects of menopause or even delay menopause. So that's the hope. We're trying to identify specific species that are able to have that effect, that positive effect on the recipient.
Yeah, we're really at a point where we don't know what the beneficial microbiota are for anything really in our gut, right? And it's such a complex, it's not just, probably won't just be one, right? It'll probably be some specific complex mixture of microorganisms.
Exactly.
I think we have time for one last question.
Yes, I'm also aging for another question, but you go ahead first, Jennifer.
Oh, I just want to know, so the microbiota can regulate estrogen metabolism. Does it also regulate metabolism of other steroid hormones that we care about with respect to female reproductive biology?
We're on the same wavelength, Jennifer.
That's something that I haven't looked into, so I can't really answer that question.
Okay. Well, maybe you could sometime in the future.
Yeah. So there's a future working plan, because I would like both our speakers today to really sort of, could you suggest, something that could be a potential helpful diet, giving the signs that at this moment, we know that there's a lot more to be done. But, sometimes we really need to tell the ladies what to eat, and they really want something that's more natural, something that could, they could be empowered, right, Lynae? Ladies like to be empowered. They like to be, know what they can choose, what they cannot choose, help themselves early on. So could you both suggest something?
I could suggest that two things. I think that, my own physician has said a plant-based diet is a really great diet for many reasons. Mm-hmm. But I think the reason that I tell patients that a plant-based diet is a good thing, and I try to adhere to it as well, is that because a lot of fruits and vegetables are enriched with antioxidants, and supplements are great, but obviously supplements may not be regulated. You may not know how much you're getting of a supplement, but if you're eating a healthy diet that is balanced, that is something that is really going to help improve, overall health. And the other thing is that heart disease is the #1 killer of women in the United States. And so what people don't realize is that there's a lot of processed food in the Western world. So meats are processed, things that you buy in cans that are not fresh. All those things have some type of processing, and they can have additives that make it have increased fatty acids and therefore perhaps free radicals. And so, decreasing processed foods and I think is really important. And then the last thing is tracking. I was able to lose, 15, 20 pounds just by tracking what I was eating because of the maternal programming that I have. My mother has diabetes. I have pre-diabetes. I was born a shoulder dystocia baby at 10 pounds. So I track, how many carbs I eat a day and try to keep a healthy, active lifestyle as well. So I think that's medicine that I take too.
That's amazing. Yes, thank you. Mary Zelinsky has a quick question. She's pointing out and asking, aren't there current studies that look at at older oocytes and exposing them to antioxidants prior to IVF having beneficial outcomes? I think you might have mentioned that, but maybe that was just in the
I
back of my
probably
head.
mentioned it for like a half a millisecond. Thank you, Dr. Ziklinski, for pointing that out. So Kelly Moley, who was a mentor of mine, still is a mentor of mine, did some of this where she gave resveratrol and I can't remember the other one right now, but she basically showed that there was improvement in like spindle morphology. And, you know, spindle morphology is important, obviously, for the oocyte because there's a lot of aneuploidy, which leads to the miscarriage. And so, yes, that is the case, but it's not something that's been implemented clinically. So at the retrieval, there's nothing that we are doing to the oocyte to prepare it or make it enhance it or make it better. There was some data from a lab group that tried to transfer, you know, mitochondria from stem cells, but there was a lot of controversy surrounding that because these stem cells could not be duplicated. So I would say right now, there's nothing that we do. And unfortunately, in IVF in general, there's a lot of take this supplement. It doesn't hurt, right? But there's not a lot of data that I would say I'd feel comfortable saying that this is in human population, that this is something that we do standardly.
Mm-hmm. Yeah. Even autologous, there's a trial recently, I think the name they may have known from Labata's group. They try to do autologous, mitochondrial transfer in aging oocytes, but that human trial didn't work out. It didn't really, rejuvenate the oocytes. So there's still more to learn about. I mean, this is exciting. We have such a long, discussion as well. Jennifer, back to you.
Yes. Yeah, thank you to both of our speakers. That was really wonderful. We're actually going to, like in previous weeks, we'll continue the discussion. If you have more questions, if you think of something tomorrow that you wanted to ask the speakers, but it took 24 hours to come to your mind, we're going to continue this discussion at gcrle.org. And next week, we have Zita Santos from Champollion Foundation in Portugal joining us, and Marco Conti from UCSF. So please tune in. And in 2 weeks, actually, Mary Zielinski will be giving a talk, who's been asking great questions all the way along. Yeah.
Yes, indeed. Thank you, Jennifer. Thank you, our wonderful speakers for today. And until next week, okay, next Friday, we're back again. So please, that's Singapore time, and Thursday evening for the US time. So till then, making reproductive longevity a reality. Thank you everyone for joining us.